All of the glycolytic enzymes, all of them are located not only in the cytosol, where they function normally, but also are located in the nucleus. Wow! All of the glycolytic enzymes. And 5 of the enzymes of the Graef cycle are also located at the nucleus. And they are performing really interesting functions, like they work in transcription or epigenetic modulations. So it's a really, really fascinating topic to me. It is my pleasure now to present to you to Dr. Lilia Torres Machorro, that works at the National Institute of Respiratory Diseases at the Cell Biology Laboratory. Lilia Torres Machorro was awarded the UC Nexus CONACYT postdoctoral fellowship to conduct research in the Division of Biological Sciences at UC San Diego under the supervision of Lorraine Pilus from 2011 to 2013. Torres Machorro uses yeast as a genetic model to study a gene that is affected in many types of cancer, ESA1 type 60. ESA1 is an enzyme that regulates chromatin, the DNA protein structure that contains the genetic information of the cell. Her work explores conditions that improve the fitness of ESA1 mutant cells, specifically when responding to DNA damage, a pathway that, when impaired, can lead to cancer. Her results describe the reasons why ESA1 mutant cells are incapable of an efficient repair of damaged DNA. Importantly, she also found different ways to solve ESA1 mutant cell repair problems by improving their ability to ligate broken DNA. These findings may have important implications for clinical research directed toward improved cancer therapies or antifungal drugs. Please come, Lilia, and give us your talk.
Good morning. Thank you so much for the invitation. It's a pleasure to be here. Today I'm going to talk about the work I performed during my postdoctoral stay at the University of California, San Diego. This work was performed in yeast. It was not aiming initially to characterize a monoiding protein. So we, as Dr. Miranda said, my lab was specialized in genetic interactions with ESA1. And we found a protein that interacts with ESA1 that is a monoiding protein. And then it's when we decided to see what happens with this protein. So first I'll introduce ESA1 and then I'll talk about isomony. Okay, so post-translational modifications are very important to regulate many cellular processes, and there are many types of post-translational modifications. These include acetylation, methylation, phosphorylation, and ubiquitination, among others. And these modifications regulate, for example, gene expression, replication, repair, recombination, lots of things. So in the, I need to look at the slide. So, yeah, that's better. Okay, so in this talk I'm going to focus on histone acetylation. This is established by histone acetyl transferases and is removed by histone deacetylases. ISA1 acetylates histone H4, and this mark is usually removed by the histone deacetylase RB3. In humans, there's a homologue called TIP60, which the same as ISA1 structures the NUF4 histone acetyltransferase complex. TIP60 is very important in humans. Well, it's essential in both organisms, and it is implicated in many diseases. For example, it is dysregulated in cancer or in some neurodegenerative diseases. Now I'm going to talk briefly about the model we used. Saccharomyces cerevisiae is an exagenetic model. So we used three of the tools of Saccharomyces. One of them are gene deletions, which is just replacing the I was just trying to make this work, but I don't think it works. Is it? Okay. Okay, so the replacing a gene with an antibiotic resistance marker, for example. The other thing are hypomorphic alleles. So in the case of ISA1, as it's an essential gene, we have to study with hypomorphic alleles. So one of these is ISA1414. This is a point mutation that generates a frame shift, and this protein loses 20 amino acids in the C-terminal region. And so it generates a partially inactive protein that can be further inactivated by growth at high temperatures. And the last thing are suppressor genes. So for this concept, I can better explain with this figure. So in here, the wild-type strain looks just fine. The mutant one is purple, so it has a problem. And then when there's a mutant one combined with mutant 2, the cells look more similar to wild. And this is considered to be a suppressor. Well, mutant 2 is a suppressor of mutant 1. These phenotypes can be better observed by growth assays. So in these experiments, the cell cultures of the different strains are normalized to a specific LD, and then they are diluted and plated on different plates, depending on what we want to test. So for example, for DNA damage, we would plate on a plate that has a drop that induces double-strand breaks. And so the wild-type strain would grow just fine. The mutant one would have some problems, and the mutant one combined with mutant 2 would grow better than mutant 1, but still not as good as wild-type. So this is how a suppression would look like. So going back to ISA1, it has many phenotypes. The ISA1414 strain, for example, is sensitive to DNA to temperature sensitivity. And in here, in a plate that is grown at 37 degrees, the ISA1414 cells cannot grow. This strain is also sensitive to DNA damage. And in here, in a plate that has camptothecin, which is a drug that induces DNA double-strand breaks, the cells have a hard time growing too. This strain has low acetylation levels in histone H4. So now we come to get to know LICE20. So we performed a genetic screen to figure out what high copy suppressors, looking for high copy suppressors of ISA1, and found that overexpression of LICE20 could suppress the DNA damage sensitivity of ISA1 in camptothecin. So the next step then was to check if the catalytic activity of Lys20 was important for this. And so we mutated aspartic acid in 155 thioualanine. This is the catalytic residue of this protein. And we call this mutant Lys20 cap. And when we did this, we found that it could still suppress the ASA1414 sensitivity to DNA damage. So the catalytic activity of Lys20 is not important for suppressing ASA1414, so it may be doing something else. So in this experiment, we had the control of a strain that could not synthesize lysine, which is in the pathway that it works. And we can see that the Lys20 cat mutant cannot grow in the absence of lysine. This is when our lab figured out that it was a moonlighting protein. So then I want to talk briefly about Lys20. So Lys20 is a homocitrate synthase. This is the first enzyme of the lysine biosynthesis pathway. And it converts 2-oxoglutarate to 3-homocitrate with acetyl-CoA as a cofactor. What was strange when it was described initially, it was that it is localized in the nucleus. And most of the proteins in metabolic pathways are mostly localized in cytoplasm or mitochondria. So people were asking what's happening with this protein that is localized, mostly in the nucleus. So in fact, you can see here the mitochondria, and it doesn't seem to be there. And this gene homocitrate, this protein homocitrate synthase is coded by two genes, Lys20 and Lys21. These are paralogs. And they have very similar protein sequences. You can see here, here there are other fungi compared to the two here. These two are Lys20 and Lys21. And you can see the dark part is like all the conserved amino acids. And in color are some areas that have been described to be important for the catalytic function. And the difference is mostly found in the N-terminal and C-terminal regions of the proteins. So we also tested if Lys21 could suppress the ASA1 phenotypes and found that it couldn't. So Lys21 cannot suppress the ASA1414 DNA damage sensitivity. So next step was, the question was, how does Lys20 expression suppress ASA1414? And the first thing I did was to try to localize the bifunctional domain. And to do this, I did site-directed mutagenesis to then do structure function analysis. And we already knew the structure of the protein. So we know that Lys20 forms homodimers, and that this green part contacts the catalytic region, which is this one, of the other monomer. And so we tested some proteins in the lead, some amino acids in the lead. This region is called the lead. Some amino acids in the catalytic region, some others in some conserved regions, and also domain in the C-terminal region that I did, a 20 amino acid region that turned out to be the moonlighting domain, as you will see. So I did the evolution assays again, overexpressing this gene in Lys20, Lys21 null strain to see if it was still able to synthesize lysine. And found that most of the mutants that I made were unable to synthesize lysine. Only, for example, the moonlighting domain, the mutant with the moonlighting domain was still able to synthesize lysine. But when we tested the ASA1414 strain for overexpression of this different mutants, found that most of them could still suppress, but the one mutated in the moonlighting domain could not suppress anymore the DNA damage sensitivity of ISA144. So this is interesting because this domain is not really conserved in any of the other yeasts. So as it happens with many moonlighting proteins, they are considered to be recent evolution, and so they are not found in close relatives of them. So the next question was, what repair pathway is affected in ISA144 mutants, and how does lysine overexpression suppresses the DNA? I'll tell you the response before telling you what, like all the experiments, because sometimes it's hard to follow without knowing what's going to happen. So what we figured is that ISA1 mutants have defects in repairing DNA damage directly at the DNA double-strand break. So these cells have, normally when there's a DNA double-strand break, there's increased acetylation at the break, and this leads to histone eviction to allow recruitment of proteins involved in repair. So in ISA1 mutants, we found that acetylation is low at the breaks, and also there's no histone eviction. When lysine is overexpressed, there's tons of lysine recruited to the breaks, and this leads to recruitment of high levels of a chromatin regulating complex that allows histone eviction normally in this strain. So this is the way we figured out, well, this is what we figured out happened with lysine. Now I'll tell you how we did it. So the first question was to test maybe overexpression of lysine was just increasing acetylation levels, global acetylation levels of the ISA1 mutants. So we needed to do this control because it's a histone acetyltransferase. But found that when lysine is overexpressed, the low histone acetylation levels in the cell remain. So this was not the way lysine was suppressing the phenotypes. Then we tested if lysine could bind chromatin because it's in the nucleus. And we did subcellular fractionation experiments where we got crude chromatin and then purified chromatin and ran Western blots and found that lysine is mostly found in the bound chromatin, same as CER2, which is a protein that is known to be bound chromatin. PGK1 was the control for a mostly cytoplasmic protein, and we can observe that there are lower levels of this protein in the chromatin fraction. When we induced DNA damage, we observed slightly higher levels of lysine at the breaks in a similar way, at the chromatin, in a similar way to CER2, which we know that it also is recruited to the double-strand breaks. Then is when we asked if lysine would be doing something directly at the double-strand breaks. So this pathway is very well described in yeast. I will just tell you a few things about this pathway. When there's a double-strand break, there's initial phosphorylation of histone H2A through two kinases, MEG1 and TEL1. Also, there are increased levels of acetylation mediated by UA4, which is the complex where ISA1 works. There are then chromatin remodeling complexes are recruited. For example, the inoidic complex is recruited and this allows histone eviction. There are other chromatin remodeling complexes like RISC1 and SWI/SNF, which are also important for opening the chromatin and allowing recruitment of late repair factors like RAD52, for example. So to test where could lysostomy be doing something, we tested if overexpression of lysostomy could suppress phenotypes of other mutants in the pathway I showed you. And found that it couldn't, for example, I included the H2A S129A mutant, which cannot do the initial phosphorylation of H2A. Or for example, TEL1, the kinase mutant, RAD52, this is a late repair factor. And we found that lysostomy expression could not really suppress other strains affecting the repair pathway. Only ISA1 was the one where we saw a clear phenotype of suppression. So we figured that the role of lysostomy should be early in the repair pathway. So to test what was happening directly at the DNA double-strand breaks, we used a reporter. So the reporter is a strain that has an homing endonuclease coded, regulated by an inducible promoter, GAL10. So when cells are grown in raffinose, this promoter is not repressed nor activated. And but when galactose is added to the media, the gene is highly induced. This leads to expression of this endonuclease that only produces a single cut in the polygenome, a single double-strand break. And then we use chromatin immunoprecipitation to analyze modification or recruitment of factors directly to the break, 0.6 kilobases downstream of the double-strand break. So it's in here, more or less. And what we first tested was recruitment of ISA1 to the break. So this was already described. ISA1 was recruited to the breaks. And we had our control no-antibody chromatic immunoprecipitation, vector transformed strains, which are wild-type, and lysostomy overexpressing strains. And so in wild-type strains, ISA1 is recruited to the break 1 and 2 hours after induction. And when lysostomy is overexpressed, similar levels of ISA1 were recruited to the break. So this means that lysostomy is not suppressing ISA1 through recruitment of high levels of ISA1. Then we tested if lysostomy was recruited to the breaks and found that it is recruited 1 and 2 hours after break induction. And when it is overexpressed, there are higher levels of lysostomy recruited to the break. Next thing we tested were histone modifications. And we tested histone H4 lysin 5 acetylation and found that in wild-type cells, acetylation of H4 increases 1 and 2 hours after break induction, but in ISA1 414 cells, there's no increase. So when we tested for histone levels at the break, we found that, we know that in wild-type cells, histone levels diminish normally because they have to be removed from the break to allow recruitment of other factors. But in ISA1 414 cells, the histones remain there, they even increase a little. So this means there's no addiction. When we did the rate of histone acetylation relative to histone levels, we found a very high signaling of acetylation in wild type, but not in the ISA1 414 cells. So to further dissect lysostomy at the breaks, we used the mutants that I constructed before. The catalytic mutant that can still suppress damage of blood synthesized lysine, and the moonlighting mutant which cannot suppress the DNA damage sensitivity of ISA1. And repeated the experiment of histone acetylation and found that only the catalytic mutant could suppress the defect of acetylation, whereas the moonlighting mutant had low levels of acetylation of histone H4. And when we tested the histone addiction of histone H3, found that Lys20 cat strains looked like they were losing histones at the break as we expected, but the Lys20 mutant couldn't. And when we did the rate, we found that the signaling was normal only for the strain that was overexpressing the catalytic mutant, but not the moonlighting mutant. So we thought then that the problem was something related to remodeling complexes. And the question was which remodeling complex is important for that. So I constructed double mutants combining ISA1 with mutants in different chromatin remodeling complexes. So RISC1 affects early nucleosome mobilization by RISC, ARC8 histone eviction by the chromatin remodeling complex in O80, and SNP5 synapses and homologous recombination by the SWI-SNP complex. And then these double mutants were tested with overexpression of Lys20 to see if we could still see suppression. And we found that the RISC1 complex was not necessary for suppression, same with the SNP5 complex, the SWI-SNP. But the INO80 complex, we needed it to be there to be able to see suppression. So this means that the INO80 complex is important for this. So we tested what happens with INO80 at the breaks and found that, well, there's a slight increase in INO80 recruitment at the break in wild-type and ISA1 strains. However, when Lys20 cat is overexpressed, there's a huge increase in the INO80 recruitment at the break. And in here I had the control of protein levels of INO80 that were unaffected by overexpression of any of the mutants. So the next thing we tested was if INO80 was interacting directly with Lys20. And we did co-immunoprecipitation. We immunoprecipitated MICTAC INO80, which is this one, well, tons of bands in there. And tested if Lys20 would come with the immunoprecipitation of INO80 and found that it did indeed, independently of if the moonlighting domain was there. So the interaction between Lys20 and INO80 is independent of the moonlighting domain. However, we found that the moonlighting domain is important for recruitment of Lys20 to the DNA double-strand breaks. We did this chromatin immunoprecipitation and found that the Lys20 cat mutant is recruited normally to the breaks, but the moonlighting mutant is not the one that doesn't have the moonlighting domain. So, just to summarize what happens in wild-type strains when there's a DNA double-strand break, histone secretion increases and histones are evicted from the break. INO80 and Lys20 are recruited there at some level. In ISA1414 mutants, histone acetylation is low and also histones remain there. And when Lys20 is overexpressed, tons of Lys20 are recruited to the break. These interact with INO80 and promote high levels of recruitment of INO80 to the break and normal histone eviction. And this allows the cells to repair normally. Just to finish, I'd like to talk about the regulation of Lys20. So when I was working with this protein, I found that its protein levels are affected very easily by many factors. And interestingly, DNA damage doesn't seem to affect at all the protein levels of Lys20. but we have some preliminary data showing that the post-translational modification of lifespan through acetylation is increased when there is DNA damage. So it could be that a post-translational modification of lifespan what is regulating that. However, when there's lysine in medium, protein levels of lysine increase. Also, we had a superoxide dismutase mutant which has high levels of superoxide and found that lysine levels are very high in this strain. However, when you grow yeast for a long time, the lysine levels are reduced the older the culture is. And lysine levels remain the same, so it is unaffected by cell density. Also, Dr. Alicia Crooks described that carbon source is also important for regulating lysine protein levels. And just to conclude, I think that lysine could belong to a backup repair pathway, so we were not able to see that it was doing something at the break at the beginning because there was no problem with repair at all. But if we have some problem with repair, then we are able to see it. However, and I think that the environmental factors are important to make the protein decide if it's going to help or not repair, depending on, well, it will affect its protein levels, and when it's low, it's not going to do anything to help repair. And well, there are many things to do with this protein, but some things that could be done is to test how its stability is regulated, how is its post-translational regulation, and how does lysine and INOAD interact, for example. And just to finish, I'd like to thank the Pills Lab and also the fellowship that I got during my postdoctoral stay. Thanks. You're welcome. The domain? No, it is not. Well, it doesn't mean that it may not be doing something else, but here in the sequence comparison, this part, even though this part is long also in IS-21, you can see that all the residues are different between IS-20 and IS-21. So the moonlighting domain is totally different.
So it would be nice to find out whether the autonomous of this gene in lactis has the moonlight property.
Yeah, I don't think I have the comparison with lactis here. Yeah, so it will be interesting to test. In fact, as when I was trying to publish this, you have to find something that is important for everyone, and so this is not the case. But we found that structurally, this region in Cryptococcus may be similar in some way, well, just structurally, because this is an intrinsically disordered region, which is also a characteristic of moonlighting proteins, well, because they are flexible, and so they can bind many things or form different structures depending on example, the environmental conditions.
Constructive evolutionary history, maybe philoxenia, and trying to see the cataracts, which it looks like a ancestral type has developed.
Yeah, that would be really interesting. Yeah, so I blasted this sequence to see like what could I find, and it showed some homology to the slingshot protein phosphatase. However, it's not similar to the catalytic region of the phosphatase. But these phosphatases are involved in regulation of actin. And actin is a very important subunit of these chromatin remodeling complexes. So I was thinking maybe it could be doing something with actin or actin-related protein in these complexes. Yeah.
¿Más preguntas?
I'm interested in the relationship between the let's say, and this one, because I read in your paper actually that this 21 is also binds to DNA, like you know that the functional form is not like also in the nucleus, binds to DNA, and that could suggest that it can also have a role. And then you also mentioned in the paper that this 21 suppresses the phenotype of it is a one, but in a lower degree than this 20. Can you talk a little bit more about that?
Well, I think that what happens is that LICE21 can form heterodimers with LICE20. And so so when we did the initial overexpression experiments, LICE20 was still there. So you have endogenous LICE20 and overexpressing LICE21, and so the heterodimer could be just doing something there because there's tons of LICE21 in there. But I think that LICE21 in fact is just doing that because it's working with LICE20, but not. I don't think it would be able to do it by itself. I don't remember if I did the experiment, but it's because it's just like a LICE20 null and I'm expressing LICE21. I think I did it. I don't remember. So.
And do you think we could try it like in the double mutant and expressing LICE21?
Yeah, that would be the thing too.
Each organisms have parallels of license. You mentioned already Bludero and Saccharomyces, but I mean, in an attempt to understand if the moonlighting activity is a result of the replication rather than a function of the original gene that has the lysine biosynthetic activity. I would think in organisms that do not have any duplication of this gene, if they still maintain the domain that you found to be the moonlight.
I didn't find any other organism, and in fact this pathway is not conserved, for example, in humans, so we cannot synthesize lysine. This is a specialized pathway of yeast and some bacteria. So
that bacteria won't have duplicated this gene.
No,
And do they have the moonlighting domain?
I didn't find any.
Any other question? Thank you.